Welcome back to the Biology Domination Podcast, your daily audio weapon for NEET 2026. I'm your host, and today is Day 7, Anatomy of Flowering Plants. Now, before you zone out, I need you to hear this. This chapter has appeared in NEET every single year, not sometimes, every year. It gives you direct diagram-based MCQs, comparison questions, and trap statements that look right but are completely wrong. Today, we are going to go through this entire chapter, stems, roots, leaves, and secondary growth, in a way that sticks. No rote learning, pure understanding. And at the end, I'm going to rapid-fire the top NEET traps, so you walk into that exam hall with 0 blind spots. Let's begin. Let's start with the stem and the comparison that NEET absolutely loves. When you cut across a dicot stem, think sunflower, think any typical herbaceous plant, and look at it under a microscope, you'll see vascular bundles arranged in a distinct ring pattern. Each bundle has xylem on the inside and phloem on the outside, and sandwiched between them is a strip of cambium. Because cambium is present, these are called open vascular bundles. And here's the consequence of having cambium. The plant can undergo secondary growth. Cambium keeps dividing, adding new xylem inward, new phloem outward, year after year. This is how a thin sapling becomes a thick, woody trunk. Now switch to a monocot stem, sugarcane, maize, bamboo. Cut it across, and you'll immediately notice something different. The vascular bundles are scattered everywhere throughout the ground tissue. There's no ring, no pattern. And critically, there is no cambium between the xylem and phloem, so these are called closed vascular bundles. No cambium, no secondary growth. A bamboo grows tall, spectacularly tall, but it never develops a thick woody trunk. That's anatomy at work. This one comparison, open versus closed, ring versus scattered, secondary growth versus no secondary growth, has generated NEET questions year after year. Lock it in. Let's go underground, root anatomy. And we're going layer by layer, from outside to inside, because NEET loves asking about specific layers. The outermost layer of a root is called the epiblema, not epidermis. That word belongs to the shoot system. The epiblema is single-layered and produces unicellular root hairs, tiny tubular extensions of individual cells that massively increase the surface area available for water absorption. And here's a critical point. There is no cuticle on the epiblema. A waxy cuticle would block water from entering. Roots need to be permeable, so no cuticle. Moving inward, the cortex. This is several layers thick, made of parenchyma cells with intercellular spaces. Water moves relatively freely through here. Then we hit the most important layer in the root, the endodermis. This single layer of cells has a very special feature, Casparian strips. These are bands of suberin, a waxy, waterproof material that run around each endodermal cell like a belt. Because of these strips, water cannot pass between endodermal cells. It has to pass through the cytoplasm, through the living part of the cell. This forces every molecule of water and every dissolved mineral through a living checkpoint. The plant gets total quality control over what enters its vascular system. Brilliant engineering. Just inside the endodermis is the pericycle. This is usually one or a few layers of parenchyma cells, and it has 2 major functions. Lateral roots originate here. They push out through the cortex and epiblema. And in dicots, the pericycle also contributes to secondary growth. At the very center, vascular tissue. Xylem is arranged in a star pattern. This is what NEET diagrams show. The arrangement is X-arc. Proxylem forms on the outside, metaxylem on the inside. In dicot roots, there are 2 to 4 xylem strands, diarch, triarch, or tetrarch. Phloem alternates between the xylem arms, and the pith, absent or extremely small in dicot roots. Now, dicot root versus monocot root. This comparison is a direct NEET MCQ source, and it has 3 key differences you must know. 1. Xylem strand count. Dicot roots have 2 to 4 xylem strands, diarch, triarch, tetrarch. Monocot roots have 6 or more. This is called polyarch. If a question shows you many xylem strands, think monocot. 2. The pith. In dicot roots, pith is absent or extremely small. The center is mostly occupied by xylem. In monocot roots, there is a large, well-developed pith at the center. This is one of the clearest visual differences in a diagram question. 3. Secondary growth. Dicot roots can undergo secondary growth through the pericycle and vascular cambium. Monocot roots cannot. Their pericycle is only a single layer thick, and there's no mechanism for lateral meristem activity. These three differences, strand count, pith, and secondary growth, will appear in NEET. I promise you that. Now let's look at the leaf, the organ where it all comes together. Cross-section of a typical dicot leaf, think mango, neem, rose. And here's what you see, top to bottom. At the very top, the upper epidermis. It's a single layer of cells, and it's covered by a cuticle, a waxy layer secreted by the epidermis. The cuticle's job is to prevent water loss from the top surface. As a result, there are very few or no stomata on the upper surface of most dicot leaves. Below that, palisade mesophyll. These are the star performers, long cylindrical cells packed tightly together, standing upright like soldiers in a row. They are absolutely loaded with chloroplasts, far more than any other leaf cell. This is your primary photosynthesis zone. The tight packing maximizes the number of cells exposed to incoming sunlight. Below the palisade, spongy mesophyll. The architecture completely changes here. Cells are irregularly shaped, loosely arranged, and there are large air spaces between them. These air spaces are connected to the stomata and form the gas exchange highway of the leaf. Carbon dioxide comes in, oxygen goes out, water vapor exits during transpiration. It all happens here. At the bottom, lower epidermis. This is where most stomata are found in dicot leaves. Each stomata is flanked by two guard cells, kidney-shaped in dicots, dumbbell-shaped in monocots, that control opening and closing. Running through the middle of this whole structure, vascular bundles. Xylem on the adaxial side, meaning facing the upper surface, and phloem on the abaxial side, facing the lower surface. Dicot leaves are dorsiventral. They have distinct upper and lower surfaces with different structures. Monocot leaves, like grasses, are isobilateral. Both surfaces are structurally similar. And in monocot leaves, the mesophyll is not divided into palisade and spongy. It's uniform. If NEET shows you a cross-section with clear palisade and spongy differentiation, that's a dicot leaf. Secondary growth. This is the chapter's most complex topic, and it's also one of the most satisfying to understand because it explains something you've seen your whole life: why trees have rings, why wood exists, and why bark looks the way it does. Secondary growth happens only in dicots and gymnosperms. It's driven by two lateral meristems, two cylindrical sheets of dividing cells that add girth to the plant. The first is the vascular cambium. In dicot stems, there's already cambium inside each vascular bundle, called fascicular cambium. During secondary growth, the cells between the bundles, interfascicular parenchyma, dedifferentiate and become interfascicular cambium. Together, fascicular and interfascicular cambium form a complete ring of vascular cambium around the stem. This ring now divides. Cells pushed inward become secondary xylem, wood. Cells pushed outward become secondary phloem. Year after year, more secondary xylem is added, making the stem progressively thicker. But the rate of growth isn't constant. During spring and early summer, when water is abundant and temperatures are favorable, the cambium produces large diameter vessels. This is springwood or early wood. It looks light and occupies more space. During late summer and autumn, when conditions are harsher, the cambium produces narrow diameter vessels. This is autumnwood or late wood. It looks dark and denser. 1 layer of springwood plus 1 layer of autumnwood equals 1 year of growth. These pairs are what we call annual rings, and by counting them, you can determine exactly how old a tree is. Dendrochronology, the science of dating trees by their rings, is built entirely on this principle. Over many decades, the innermost xylem, the oldest wood, stops conducting water. It fills up with resins, tannins, oils, and gums. It becomes dark, dense, and hard. This is heartwood, also called duramen. It's the structural core of the tree. Beautiful for furniture, useless for water transport. The outer xylem, still young, still functional, continues conducting water and minerals. This is sapwood, also called alburnum. It's lighter in color and is the tree's active plumbing system. The second lateral meristem is the cork cambium, or phellogen. As the stem expands due to secondary growth, the original epidermis can't keep up. It tears. Phellogen forms in the outer cortex and takes over. It produces phellum, cork cells, on the outside, and phelloderm on the inside. These cork cells are dead at maturity, with walls impregnated with suberin. They form an excellent waterproof insulating bark. Phellogen plus phellem plus phelloderm together form the periderm. This replaces the epidermis as the protective outer covering of old stems and roots. NEET has asked about heartwood versus sapwood, springwood versus autumnwood, and the origin of cork. Know all three cold. All right, it's rapid fire time. This is the segment where I say a statement and you decide true or false. Do this out loud if you can. Active recall is how memory forms. Statement 1. Monocot stems have open vascular bundles. False. Monocot stems have closed bundles. No cambium, never open. Statement 2. Casparian strips are made of cellulose. False. Casparian strips are made of suberin, a waxy material. Do not confuse this with the cellulose cell wall. Statement 3. Palisade mesophyll is the site of gas exchange in the leaf. False. Palisade is for photosynthesis. Gas exchange happens in spongy mesophyll thanks to its large air spaces. Statement 4. Sieve tube elements have a well-defined nucleus. False. Sieve tubes are living, but enucleate. They have no nucleus at maturity. Their companion cells have the nucleus and control their activity. This distinction has been directly tested in NEET. Statement 5. Heartwood is the functional xylem that conducts water. False. Heartwood is non-functional. It's the dark, resin-filled core. Sapwood is the functional, water-conducting xylem. Statement 6. Lateral roots originate from the endodermis. False. Lateral roots originate from the pericycle. This is called endogenous origin. They form deep inside and push outward. 6 traps, 6 marks potentially saved. And that's a wrap on day 7. Today you went inside the plant, deeper than most students ever bother to go. You understand stem anatomy in dicots and monocots. You know every layer of a root, and why the Casparian strip is the plant's most clever adaptation. You can read a leaf cross-section like a map, and you understand secondary growth, the process that builds trees, creates wood, and leaves annual rings behind like a biological diary. This is not just memorization. This is understanding. And understanding is what gets you marks when NEET gives you a twisted question you've never seen before. Tomorrow, day 8, we move to structural organization in animals, with a special focus on the frog, a brand new addition to the NEET syllabus. Don't miss it. If today's episode helped you, share it with one friend who's also preparing for NEET. That's all I ask. All notes, PYQs, and resources are linked in the description. linktree.tree/hiddenwisdomacademy. I'll see you tomorrow. Keep going.